Sorry, let's start recording again. And we're back. So, one of the early methods for showing genetic influences on behavior are comparing the behaviors of inbred strains. So these are strains that are going to be genetically distinct from each other. Initially, just looking at random variation among individuals, but then when you create inbred strains, you can, in a controlled, replicated way, look for differences in behavior, where the only difference is that the strains are genetically different from each other. So in this study by McCloskey, 1997, paradise fish, which are beautiful and are bred by breeders for their color, and so you often have different varieties, inbred lines. So the whole point about inbreeding is that whatever genetic variation you have over several generations of inbreeding will be lost, and you'll have be homozygous at every locus. But if you inbreed several different lines, they'll end up through drift facing for different alleles at different loci. And what McCloskey found was that they were able to isolate lines that varied in anti-predator behavior. In other words, paradise fish are sensitive to eyes. That's what they're like looking for in a predator. And so if you present them with a model that doesn't have eyes, then neither the P line or the S line flee. And so let's take a look here. So this is without eyes, no difference. And what's the dependent variable? The dependent variable is swimming away, fleeing. That's our dependent variable. However, when you present them with a model, and that means a fake fish, like a pike or something, if the model has eyes, then the P strain flees. However, the S strain never shows an anti-predator response, right? So because you're looking at different inbred lines and everything else about the experiment is the same, what this shows is that there's a gene, at least one gene, that affects the ability to respond to predators by recognizing eyes. Yeah.
Professor, I'm a little confused about the S and P strain. What do you mean by, I understand the response to the predator with eyes and without eyes. However, when you say strain, what do you mean by that, P strain and S strain?
So it means you start with a large outbred genetically diverse population, and you randomly select small numbers of individuals, like pairs, and then you inbreed the offspring of those pairs. So you mate them to each other. And this is going to do a few things. One is any really deleterious, that means harmful, alleles that would only be expressed as in recessive form get brought up. And so you have high mortality, and sometimes those lines die. But after you've done inbreeding for a few generations, two things happen. One, you get rid of some of those deleterious recessive alleles. And two, loci that might have been heterozygous initially, after several generations of inbreeding, you lose any genetic variations, so that you end up with strains of individuals who are genetically identical and also perform pretty well in breeding, because you've gotten them over the hump of inbreeding depression, right? Like most animals avoid inbreeding because presumably ultimately because inbreeding expresses any deleterious recessive mutations, and we all carry a bunch of them. But if you force them to inbreed, basically end up getting rid of those, but then you also end up losing all genetic variation. So if you do that to a bunch of pairs, you'll end up with multiple lines that are different from each other genetically because there was originally genetic variation, and then within each line they become homozygous, right?
Oh, okay.
And then so there wasn't like selection, and in this case there wasn't like induction of mutation. We'll talk about that later with Drosophila.
Mhm.
um, but you just have lines that are homozygous internally but different from each other, and then you test them with a bunch of behaviors.
Okay, that makes sense.
Yeah, it's like it's an early technique for looking for whether genetic variation in a population can lead to meaningful behavioral changes. um, this is another study. This is a cross-fostering study. And in some ways, this is looking at different species of voles. Um, so they normally they don't mate with each other, but they interestingly they differ from each other behaviorally. So prairie voles are monogamous; they stick together as pairs, and they have male parental care. Um, and they're more socially affiliative in general. Meadow voles live solitarily. The males and females get together just to mate, and then the males don't contribute to parental care at all. And the interesting thing, so this has been studied, a lot as a model species by I'm trying to think, Benny McGuire is, that's where the photo is coming from, and a bunch of her group is where a lot of the studies came from. But it's been studied by a few labs because maybe the question being like, is this a model for like human monogamy? And they studied genetic and hormonal correlates of pair formation. Um, in this example, if you cross foster pups. In other words, if you take meadow voles, which normally don't show any paternal care, and you foster those meadow vole pups into prairie vole families, so they are reared by prairie voles, they end up showing increased parental care. They end up taking on the behaviors of the prairie voles. And this is really interesting, and it's probably related to some of the effects of like similar to the high licking and grooming versus low licking grooming behaviors in rats, which we're going to talk about a little bit later in the course. Another way of seeing if genes are influencing behaviors by doing a selection experiment, right? So you do artificial selection the same way you would do artificial selection for, milk production or, um, you know, time to producing seeds and corn. Um, you do selection for a behavior. And the idea is this: if you do artificial selection and the trait doesn't change over time, then that means that that trait is not being affected by genes, right? It means that there was variation among individuals, but it was caused by the environment, because when you only let the individuals with, the highest level of that trait or the lowest level of that trait reproduce, you end up with offspring that were the same as the population average before. So if you do selection and you get no response, that shows that genes are not affecting that behavior. But if you do selection in a controlled environment, and then you do get a change in the value of the trait in the offspring relative to the population before, then genes are affecting that trait, because there has to be genetic variation for to have a response to selection. So a really classic example is research done by Cheryl Lynch on nest building in mice. There's a great system because a lot of the challenge with doing animal behavior is coming up with good operational definitions. In other words, there's a behavior that you're interested in. You have to figure out, how can I measure it objectively, in order to do science. And sometimes the challenge is, the way we're measuring the behavior like either interferes with it or doesn't have external validity because it doesn't translate from in the lab to outside. But this is a really great trait. um, you rear the mice and you give them, enclosures, an area where they can either forage for food and collect nest material, and you just weigh the amount of nest material that they put in their nests. And that's a great, super repeatable, unbiased measure of the behavior, which is nest building. But you measure it by weighing the nests. And so I said Cheryl, it's Carol. What Carol Lynch did, over like a 2 or 3 year study, is she had 6 lines. And initially they were drawn at random from, a large population. And 2 of the lines were high. They were only the 10% heaviest um, nest um, individuals um, were used to breed, right? So they'd have these mice and they had free range in an area where they can forage for food and nesting material, and then they'd weigh the nests that they made. And so in those high lines, they would only take the individuals that made the 10% heaviest nests. Similarly, the 10th so the 90th percentile and above of nest weight. In the low populations, they took the 10th percentile and below. And in these controls, they just grabbed 10% of the population at random and used those as the sires and dams for the next generation. And, hopefully we're getting better at looking at graphs. The first thing you want to do is look at generations. So this is a pretty substantial study because it takes, I think, at least 3 years, 3 weeks, not 3 years. Mice are awesome. Like 3 weeks to go from pup to pregnant for mice. So this is like a 2.5 year, almost a 3 year study, because they're doing 48 generations. Um, and on the y-axis, we have the grams of cotton. And you remember that that was our operational definition. You just weigh the nests, which is awesome. And it's really interesting. There's a bunch of interesting things about this graph. One is you see a really big jump here, right, in the first like 10 generations. And then oddly enough, it goes down, but then it's up, and then it's burbling around. Maybe it's going up again at the end. I'm not sure. This is actually um, a pattern that you would expect to see, and if you smoothed it out, it might look something more like this. Why might we expect to see? And in the low lines, basically you end up after, 30 generations, they don't collect any nest material. You've just gotten rid of that trait. But I'm gonna throw this question out in chat. Why might we expect an initial strong response to any kind of selection, but artificial selection Mm. It's a pickle. Because all individuals selected are similar, but the offspring becoming more diverse. Well, I actually think the opposite happens in selection, right? You start out with a genetically diverse population, you're only letting 10% breed, and so each generation you're getting rid of the genes for in the high lines for low nest building. So that's totally possible. That's not what I think, but that is possible. Um, I mean, it looks like over here you can have a generation with 50, but from generations like 50 grams, right? So it's possible they can do about 50 grams. I've lost the ability to draw a straight line, but they seem to be burbling around at like 35 to 40 grams. So I'm going to say, although that's possible, I don't think that's what's going on here, but it might be. The fact that you got this data point argues against that. The later generation offspring lost the gene. So what genes did they lose? So I would argue, no, they still are nesting, right? So like these guys are are nesting in. Whoa, sorry. These guys um, are producing nests, and they're gathering, like 20 or 30 grams more nest material than they start off with. All the populations start out around 15 grams, and now they're averaging like 40 grams. So equilibrium between what? Yeah, maybe like there's some other constraint. So Kaylee is saying the amount of nest material is a constraint. You might be implying carrying capacity. They're not competing with each other. Actually, that's a good idea, but no, I don't think so. I think there's something much simpler going on. These are small populations. It's a lab study, and that means let's say there's 100 individuals in each line and only 10 breeding. That's not many chances for like new mutations that might affect um, uh, nest building. So what happens is when you do selection, selection is incredibly powerful. You get rid of all the genetic variation for like in the low nest building direction, and then you hit a constraint because all you're stuck with are the genes from the individuals that made the biggest nests to begin with. And it is entirely possible that if you did this study either for many more years, giving many, many more opportunities for new mutations, new genes to arise that might increase nest building behavior further, or if you did this study on a large population, you might well end up with much higher nesting material. The reason why like my mind immediately goes to this is because you see a strong effect in my line of work, which is studying the evolution of resistance to pesticides, that when you do selection in the lab on small populations, you always get an effect on resistance. In other words, you do selection and you'll increase the average dose of pesticide that your population in the lab can support. But you almost never get individuals in a lab selection study as resistant as the populations that evolve in the field. Because in the lab, you have populations in the hundreds, maybe thousands, depending on the species. In the field, you have populations of millions, 10 millions, 100 millions. And that just means many, many more opportunities for new mutations to arise. In a lab study with small populations and short numbers of generations, you end up just getting rid of all the variation for the trait, which is kind of interesting. The other thing that's really interesting about this study is you can see also that in the control lines, whoa, sorry, in the control lines, it looks to me like nest building goes down. Why might that be happening? And it looks like it's a real trend. It doesn't look just the variations, not just bouncing up and down. I mean, it makes sense in the low lines where they're only allowing the lightest nest builders to breed, and it happens faster in those. So genes definitely affect nest building in mice, and we'll see a couple more examples that aren't in the text. But what's going on with those controls? Fatigue. These are multiple generations, right? So each one of these are like they're babies, they're full of energy, and all these mice are young. So yeah, I don't think it's fatigue because these aren't the same individuals, right? Each one is a new generation. It could be random drift. It could absolutely be random drift. But what's interesting to me is, and that's why she did she did the minimum, but she did 2 lines in each treatment, high, control, and low. If it was random drift, I would have expected like one control line to increase and one control line to go down. It's tough because it's only 2 lines, but both of them declined. Um, so I actually suspect that the lab is different than the field, and these mice were all being kept probably in an environment where temperature was maintained at a comfortable level. And if that's the case, if there's any trade off in nest building, like if the mice that collect more nest material spend less time collecting food, or getting ready to mate, then maybe there's mild selection against nest building in the lab, and that's my guess. But you need to do a follow up, right? You need to replicate this much more than 2 times, and you maybe would need to do an experiment where you bred mice without selection but in different temperature environments, and you see what happens to nest building, and you see if at this temperature whether it always declines. Anyway, great study. And the fact that the lines separate based on the selective treatment shows that genes are affecting um, nest building. um, Another way of, if any of the resources are limited in the lab, yeah, I'm not sure. Um, I'm not sure if they had only a limited amount of time where they could forage for nest material and food, or if it was ad lib. Okay, another another way to look for the effects of genes on behavior is to induce mutations and see what happens to behaviors that you're interested in studying. So this is, the research program that made, I think, Seymour Benzer famous, and many, many mutations were induced in fruit flies to study all sorts of traits, including learning behavior. And so there are multiple mutations that have been identified that can reduce learning ability in fruit flies. Interestingly, I think both of these are associated initially with olfaction, with smell, but they um, at least rutabaga, seems to be associated with more than one kind of learning, which is really interesting. But they know what what the protein products do within different um, cellular pathways. So dunce fails to produce cyclic AMP phosphodiesterase. It's an enzyme that breaks down cyclic AMP, and so they have high cyclic AMP levels. Um, Rutabaga fails to produce a different enzyme, ATP adenyl cyclase, and so they characteristically have lower cyclic AMP. But both genes reduce learning ability, especially odor learning, and both genes are also involved in courtship behavior. Drosophila have been courtship behavior, the genetics of courtship behavior have been studied a lot partly because it's a really, like the ducks, it's a really nice behavior that can be broken down into many um, sub behaviors and rituals, orienting to female, tapping, singing, which is actually a vibrational sound, a vibration that's made with the wings, um, licking with the proboscis, copulation, attempting to copulate, and copulation. The courtship behavior is kind of cool. So I am going to try sharing the screen because I have a window ready. Here we go. This is what it looks like. I don't know if we can hear it. That's okay. But you can see there's orienting, and then you can see the male, the rear flies wing vibrating and holds like one wing out, and it's doing its vibrational song. And that's done at an orientation. In an arena this small, they're probably going to end up mating, but in a larger, more naturalistic environment, that you have to do a lot of singing to actually get a female to mate with you and not just fly away. Okay. And we're back. So fru, which is short for fruitless, is a single gene that has large effect on courtship behavior. Um, the fruitless gene was named because the males would, instead of orienting towards females, would court males and females equally. Um, so um, one of the things, interestingly, dunce and rutabaga, which are involved in learning, are also needed to suppress courtship following encountering a mated female. So males should preferentially orient towards females that haven't mated yet because of a first male advantage in sperm competition. Insects are really interesting. Females often have really complex sperm storage organs that can either favor the first male to mate or the last male, or be sort of democratic. Um, a whole nother set of genes that have important effects on behavior are the timing genes, the period genes in Drosophila that regulate circadian rhythms. So circadian rhythms are, internal time measure internal rhythms that regulate things like appropriate time appropriate behavior like wakefulness versus Um, versus sleep, it's interesting like what sleep means for an insect, but there are periods of higher and lower activity. Um, and the period gene expression, the messenger RNA levels, oscillate on roughly a 24-hour period. And that codes for a protein product, which then feeds back and inhibits activators for the locus. So that feedback loop times daily rhythms. And there are many, many described mutations of this gene that can either make that internal clock shorter or faster, right? So the wild type PER locus gene has a 24-hour activity, 24-hour period, um, but then you have different mutations. One that can make the day a little bit shorter each time, so they start activity earlier each day. Ones that can make the period longer, so they start activity a little later each day. Some that make the daily timing irregular, and others, much longer. So that's kind of cool. Um, And one of my favorite behavioral mutants, and this is actually one that's found. This is different. Up until now, I've been talking about Drosophila behaviors just in the Drosophila section, where mutations are induced in the lab either by radiation or by a chemical mutagen. Um, and so presumably novel mutations are being created in the lab. Um, this is an example of naturally occurring genetic variation. So if you um, go out and collect Drosophila from the wild and you collect a large enough population, you'll have some that are showing the rover behavior and some the sitter. And so this is behavior that's described in the larvae, although it ends up affecting adult behavior as well. Sitters will remain in a patch if there's sufficient food. So what we're looking at here are they're actually like petri dishes that have either just the baseline agar, which isn't very nutritious for the flies, or patches of yeast, which is what the larvae are trying to eat. You're trying to eat rotting fruit. That's what you're actually eating is the yeast, the fungi that are breaking down that fruit, that food source. And sitters, if you put them in a patch of yeast, they will stay in the patch of yeast. They won't leave it to go into non-nutritious agar. Whereas rovers, um, will a fraction of them will move even if it takes them out of a food patch, in this case a yeast patch. And so if you look at behavior outside the patch, it looks identical. If you look inside the patch, rovers are going to have um, shallower turns and longer legs, and sitters in their movement paths, and sitters are going to make more frequent sharper turns. Um, so and they're going to have shorter trails. So what you can see here is there's some overlap, but on the x-axis we have foraging trail length. Um, the sitters have an average of about 5 centimeters in the petri dish trial within a yeast patch. The rovers, it's on average like, I don't know, 16 or 17 centimeters, and that often takes them outside of the yeast patch. And that's potentially important because in time, every patch gets exhausted. And so some individuals that leave the patch are going to die of starvation, but some of them are going to find new patches and benefit from that. And it's kind of interesting if you take true breeding, homozygous rover and sitters, and you breed them, um, then you're going to get rover offspring. Rover is dominant to sitter at the foraging locus, um, which is kind of cool. It just shows that this is a single gene effect that's behaving in Mendelian fashion. Um, so one question is, is it controlled by a single gene? And the way you do that is, I mean, you mate them once, and they're all rovers. But then if you do the dihybrid cross, um, you should get a 3-to-1 ratio of rovers to sitters, and that's actually what they got in the late 80s when they did those crosses. So it's kind of cool, natural variation in the wild where you have a single gene with a very significant behavioral effect. Cool. Um, Black-capped warbler is it's a species that's been studied a lot for migration in the old world. So black-capped warblers breed all over Europe, and then they try to avoid the Mediterranean, but they migrate and then overwinter in Africa. And because they have a wide breeding range and they hate flying over water, they're trying to avoid the Mediterranean. Um, you have different populations. And so the Western Germany um, uh, black-cap warblers migrate in a south westerly direction, and that makes them, and then they overwinter in um, North and Western Africa. By contrast, um, the Austrian black caps, when you migrate in a southeasterly direction, that makes them avoid the Mediterranean on the east. They go through the Rift Valley and they overwinter, Ethiopia, Kenya, Eastern Africa. And the great thing is, this is research done by Andreas Helbig um, at like Max Planck Institute for Ornithology. If you bring them into the lab, um, it's a wonderful system because these are most birds, all the songbirds that migrate at night. The gliders, the raptors, you'll see them during the day, um, vocally lagging. Ugh, con. Let's see. I got all the bars, so I'm not sure what I can say. All I can do is try to keep going. Um, okay. So the nice thing, well, hopefully then the recording will be okay. So the nice thing is all of these birds have the migratory restlessness, the Zug und Ruhe, and you bring them into the lab, and at night you put them in a cage with a little bit of a cone with an ink pad at the bottom, and the birds will try like crazy to fly, and their feet will get the ink and it'll go scrabble scrabble on the paper that's put on the cone, and then you can see what direction they're trying to fly, and you can put these cages with the cones and the ink pads out of doors or in a planetarium, and you can study their orientation. This has been used to show how they use a celestial compass, how they use the stars, how they use magnetic fields to calibrate that celestial compass. Just a ton of great work that's been done. But what this was done was just to show that when you catch birds in the aviary, they will orient consistently with their population. So the ones from Germany, southwestern Germany, try to fly southwest. The birds from Eastern Austria try to also avoid the Mediterranean, although all of this is done in aviaries at night. They try to fly to the southeast. When you hybridize these two birds and you rear their offspring in captivity with no experience migrating, the hybrids orient in an intermediate direction. And so these hybrids reared in captivity, so no migration experience. They show a migratory orientation intermediate between the two parents. So hybridization experiment is another way of showing um, that a genetic effect on behavior. Although in this case, there's no dominance. They seem to, be somewhat intermediate between the two parents. Another species that has shown um, really interesting genetic components to behavior are orchard versus deer mice. Um, and these mice nest in, along a gradient in the south, and they nest in soils that will vary in how much silt they have, which will affect kind of how easy it is to dig. And so a mouse nest will it'll have usually, at least an orchard mouse nest, will have two parts. It'll have an entrance tunnel, and that the length of the entrance tunnel can vary. And then it'll have an escape tunnel. So the escape tunnel actually isn't open, but it's close to the surface. And so a mouse that has to try to get away from a snake can race out the escape tunnel and leave the nest. If there's a snake, I'm not going to draw the snake, but okay. Now, um, the genus, both orchard and deer mice, in Paramiscus. Paramiscus polienotis burrows vary with silt content. In other words, um, the there are some burrows that they're making in sort of high silt and some in low silt. And if the greater the silt content, I think the easier it is to dig in the soil, and they will dig deeper burrows. But here's what's really interesting. Um, we have oldfield mice or orchard mice have long entry tunnels and an escape tunnel, whereas deer mice have short entry tunnels and don't build an escape tunnel. Okay, so we have two traits associated with the nest that can be measured. And when you hybridize them, the oldfield, the orchard mouse traits are dominant. They dig long entry tunnel nests with escape tunnels. Um, when you do a back cross to the deer mouse, you will get 50-50 long versus short tunnels and 50-50 um, escape hatch or not escape hatch, right? So this is nice example of um, um, two traits, each one being recessive, if that makes sense, um, and assorting independently, right? Why are you getting 50-50 and not 3-1? Because this isn't a dihybrid cross, right? This is a back cross. So you're mating to, it's more like a test cross, right? Because the deer mouse has nothing but short length, entry length genes and no escape tunnel. And so when you're mating this hybrid, this dominant phenotype individual for both traits, you end up getting 50-50 um, escape tunnel, no escape tunnel, and you're getting 50-50 long entry, short entries. Really a nice system. This is Hopi Hookstra's lab. The interesting thing is that if you do hybridization studies and backcross, you get different different effects. So the backcross seem to imply just two loci, but when you do QTL analysis, it looks like you're getting more than just two loci. So you're finding three loci, each having an effect on entrance tunnel length, um, although only one locus that has an effect on escape tunnel or no escape tunnel. Okay, so I'm going to leave nesting behavior in mice and going to talk about foraging behavior in Drosophila. So one thing, so um, one approach to studying the effects of genes on behavior is to look at orthologs. So these are sequences that have a common ancestor, but now are found in different species. And there are many, many examples of those. I mean, some are highly conserved genes, but then others are interesting because the behavior may not be found in both species. So in other words, we looked at that foraging gene, rover and sitter, remember? So just to remind us, the foraging gene in Drosophila is rover versus sitter. But in honeybees, it has behavioral effects, but sometimes in a different context. So it will affect how long a worker spends nursing versus graduating to foraging outside the nest in a honeybee colony. Um, Similarly, it will affect, its expression will affect foraging outside the nest versus nursing newly emerged brood in red harvester ants. And interestingly, it works in the opposite direction. And then it will also affect aggregation behavior in C. elegans. Okay, so um, another approach to studying the effect of genes on traits is QTL analysis, quantitative trait loci. So this is a breeding design where you identify many markers. In other words, you use a primer with PCR that's pretty short and will kind of break up the genome into hundreds of regions, and then you do statistical associations between the lengths of those regions or the sequence of those regions with a trait. And so this is good um, for looking at traits where many genes are having an effect, which is most traits. And one of the really nicest examples in behavior is looking at pollen versus nectar foraging in honeybees. So honeybees, hopefully you know or remember the basic social system. You'll have a single queen. You'll have um, tens of thousands of workers that are all sisters, the daughters of that queen, although many of them are half sisters because the queen goes on a single mating flight in her lifetime before starting a new colony or um, taking over a colony after the old queen swarms. She'll mate with many males on that mating flight. Um, and so you can look at these entire colonies. They are a single family. They often function as a single as a superorganism. And so you can sort of test colony level traits, and that will tell you, you know, about, and then you look for the genes that are in the queen and the drones that she mated with, and look for what genes are associated with what traits. So there are some colonies, and you can do selection on this on the colony level. And over generations, you'll end up with some colonies that collect more pollen, other colonies that collect more nectar. And I want to see if I have. Um, I forgot to get this one teed up, but it's here. And I'm going to share the proboscis extension reflex with you. Okay, because it's just a great way of studying behavior. Nope, not a file. Application screen, window, proboscis extension reflex. Okay, so this is from James or Richard Nies lab. He's at UC San Diego, but it was a guy in Germany that developed it. So you can train the bees with a sugar dose and give them a smell or a color cue, and you touch the sugar to their antenna, and they will stick out their proboscis because of the sugar. And so what's great is, so that's the proboscis extension, and you can see it's a reflex. It's kind of like kicking your knee up. And the great thing is you can associate the sugar with a smell or an odor, and then you can see how quickly they can learn an association. Cool. And it's fascinating because um, what we've learned is um, how um, colony level behaviors depend on individual level traits. And in this case, it's the sensitivity to sugar. So high pollen honeybees are more sensitive to sugar. And this was really fascinating because initially this was Shiner, Page, and Erber. Robert Page is a was a sort of behavioral geneticist or ecologist at UC Davis, and what they found was that the initial discovery was that high pollen honeybees um, learn faster this using this proboscis extension reflex. And then what they discovered was, well, it wasn't that they learned faster, it was that they would learn at a lower sugar concentration. And that was shown using this reflex even without an associated odor. Um, and it's really interesting. So what happens is on the colony level, the high pollen honeybees are more sensitive to sucrose. So they learn and respond to low nectar flowers. So they're not getting nectar, but they're at least getting pollen. In contrast, the low pollen honeybees they're not dumber, they're pickier. In other words, they only learn a flower source and start increasing the foragers to that source when it has a lot of nectar. And so they end up not having to visit as many flowers, and they collect honey, but they're not going to have as much pollen. They're going to be lower as in pollen on a colony level. Really cool. So you can select this behavior. They've identified 3 loci, I think, that have an effect on sugar sensitivity and therefore pollen collection. So sugar responsiveness affects how quickly they learn and learn to associate colors or odors with that sugar reward. Um, and that sugar responsiveness then affects their foraging choices. So it's a really cool system. Um, another suite of traits that have been investigated genetically is the genes that are turned on or turned off as over the course of development. So honeybees can show what's called age polyethism. So poly, many, or different behaviors. So the general pattern is that younger bees stay in the hive, they do cleanup duty, they feed the young, they do nursing, and only after about 2 weeks of age do they start to go out of the nest to forage. And before that intermediate, they might be guarding the entrance, and then they go out and forage for nectar, pollen, and if they're in a swarm, for new nesting sites. So microarray techniques are used to assay for gene expression. So the downside of microarray data is that you almost always get a lot of hits, right? So when you're looking at, okay, what genes are being turned on right now, you might find, 1,000 genes or 500 genes that are associated with a certain behavior. Um, but if you know how those genes are working already, it can be really useful. So what happens is you see what genes are being turned on in honeybees. You then look up what those functions of those genes are in Drosophila, because that's the system where the largest number of genes have been identified and their functions identified in terms of um, transcription products, and then you go back to your honeybee data and you try to say what genes are important. So in a normal colony, um, age is confounded with behavior. In other words, you might not know whether a gene is being turned on because the bee is doing nursing behavior or gearing up for foraging, or if that gene is being turned on because the bee is young or the bee is old, right? And so in order to sort of separate that and find out what genes are actually affecting the behavior, um, researchers created colonies artificially, and it's not that hard to do, in which all the bees were the same age. So they can do that by collecting eggs from many hives and putting them all in one hive and removing the younger brood. And so then you end up having a pulse that everyone is going to be the same age for at least a week or two. Um, and in this context, partly because of the needs and the hunger for the hive, some young bees are going to end up being sort of forced into being foragers. So they'll be the same age as the nurse bees, but they will be foraging. And then you, you know, see what genes are being upregulated. They show up in yellow, and which ones are being downregulated or expressed, and those are in blue. And there are, a bunch of genes that are associated with being a young forager or an old forager. So when the bees switch from nursing to foraging, these genes are turned off, and these over here are being turned on. And what's interesting is, you can see the difference between just being old and young and a nurse versus being a young forager. So, Fatima, normally the foragers are older than the nurse bees. If you experimentally create colonies where everyone's the same age. You get rid of the old bees and you start off a cohort that's nothing but eggs. Um, then you end up with a colony where all the adults are the same age, and some of the young bees will switch to foraging, partly because they're also responsive to the needs of the colony, the presence or lack of honey. Okay. So the interesting thing is the changes in gene expression were so reliable that researchers could recognize whether a bee had been collected in the colony doing nursing behavior versus being collected returning from a foraging trip based on gene expression, which is kind of cool. And then you go and you look at the individual genes and see what their function is associated with in Drosophila. And I don't know all of these enzymes, so that's not I'm not going to be able to give you tremendous insights. So on the bright side, you get a lot of data. You can identify not just a single gene traits, but you can identify networks of genes that have to work together. And then it gives you projects to follow up on. You try to figure out why a gene that's associated with one enzyme is also affecting this behavior. On the downside, you get lots of data and it can be overwhelming. You can get sort of false positives and negatives. You have to actually do it a few times. And it's a little pricey. Okay. Um, so the other thing about gene expression is you're not sure if these genes are being turned on to cause the behavior or if they're being turned on as a result of the behavior. Like the bees are outside, they're getting all sorts of different stimuli, and that leads to genes being expressed versus the genes being expressed, so they start going on foraging trips. Um, so the other thing that's different is in QTL loci, you're looking for genes that differ in sequence, right? You're looking at different versions of genes, whereas and you're looking at loci, whereas in microarrays, you're not looking for different versions of genes. You're looking for what genes are being turned on or turned off. And for that reason, you're going to get a lot more hits, but you also don't know which of those genes are being turned on as a result versus which ones are sort of driving the behavior. Um, a way that you can tell whether candidate genes are necessary for a behavior is by doing knockout studies. And so that's now a lot easier using RNAi. So you can make double-stranded RNA that binds to an enzyme, and because it's complementary to it, and then it silences that um, messenger RNA. So it's going to stop producing more of that enzyme. And so you can introduce interfering RNA to that's complementary to the gene of interest, and that'll prevent that gene from being expressed. So one one gene that's been studied with RNAi is vitellogenin, which normally suppresses juvenile hormone. So if you take entomology, one of the things you're going to discover is that almost every sort of behavior in transition is modulated by either juvenile hormone, which suppresses molt, or ecdysone, which stimulates molt, but then also has lots of other effects in the adults after, long after molting and growth is happening. So vitellogenin, which normally suppresses juvenile hormone, will keep the bees acting as nurses. But so if you knock out vitellogenin, they switch to nursing earlier. So um, this is kind of cool. Um, so um, one important like feature or thing to think about in sort of evolutionary genetics is pleiotropy, the idea that each gene can have multiple effects, can have effects on multiple traits. And so these pleiotropic effects often actually are sort of antagonistic. In other words, they might introduce costs to a trait. Um, so vitellogenin turns out, not vitellogenin does many things. Vitellogenin um, knockout bees, they start foraging earlier. They also collect more pollen, so that means they probably are more sugar sensitive, so they're willing to learn flower sources. They also survive for a longer period of time. So it's not that either of those traits are costs, but there are other traits that are associated with the same gene. Um, so knockout mice have been used to study monogamy in those prairie versus meadow voles. So breeding knockout mice is pretty easy. Um, and the interesting thing is if you knock out mice, they will lose the function of gene of interest through development and as adults. However, you can also create transgenic mice where um, um, a gene is guaranteed to be expressed. So in other words, instead of knocking out the gene and looking for a loss of function, you can create a mouse, a transgenic mouse with a strong promoter to increase the expression of a gene as well. And that's really important because it turns out that the differences in behavior shown by meadow and prairie voles um, don't depend on the differences in the gene for it's going to be vasopressin that's coming up. Um, it's going to turn to the sensitivity or the promoters for that gene. Okay, so remember, prairie voles have more parental care, they are more gregarious, and they mate monogamously, at least for a season. Whereas montane meadow voles are solitary and less social. So vasopressin is a hormone that has many effects. It has many behavioral effects. It affects aggression and parental behavior, and also pair bonding and affiliativeness. So obviously a good candidate to differ between um, these two species. Um, there are two different vasopressin receptors, V1A, V1B. So these are areas in the brain that are excited by vasopressin. Um, the interesting thing is that prairie voles and meadow voles are almost completely identical. What's different between them is the promoter region that are is greater in the Microtus ochragaster in the prickles. So the genes don't differ, but the promoter region is larger. And so what you can do is you can take the prairie vole promoter region and you can put it into meadow voles, or you can put it into mice and see what the effect of having the different promoter is. So what having the prairie vole promoter does is it regulates the transcription of the V1A RNA and then the amount of V1A protein. So what you have here are wild type mice, prairie voles, and wild type mice that are transgenic with the prairie vole V1A, vasopressin promoter in them, and they end up showing vole-like expression of vasopressin receptors in the brain. So you can see the arrows, the 3 arrows over here. In that case, the mice don't have any vasopressin receptors in these 3 brain nuclei, but both prairie voles, that's the solid black, and transgenic knocked-in mice with prairie vole V1A promoters do produce these vasopressin receptors. So it's really interesting, right? There are lots of different reasons why their behaviors could have varied, and a lot of them turned out negative, right? You could see maybe they differ in the amount of vasopressin that's produced. No. Maybe they differ in how much vasopressin, how quickly vasopressin is broken down, all those enzymes that might break down this enzyme. No, that's not it. They differ in vasopressin receptors, how sensitive they are to vasopressin in different parts of their brain. And that's not because their genes differ for vasopressin receptors, but because of the promoters that code for those receptor genes differ. So you can, with more modern techniques, you can really identify mechanisms underlying genetic variation effects in behavior. So if you just increase the receptor, that's not going to have an effect on behavior. But if you also have vole level vasopressin in the transgenic mice, you end up affecting their behavior. So just because you get mice with vasopressin receptors in their brain that they normally don't have doesn't necessarily mean you create prairie voles out of mice behaviorally. You also need the prairie voles also, and the meadow voles both probably have higher production of vasopressin as well, but the prairie and the montane meadow voles differ in how they respond to them. Okay, it's usually my goal to not go till the very bitter end, and I started early enough, so I am going to stop here. And I'll just finish this up next time that we meet. And I'm going to talk more about the paper and paper topics next time that we meet as well. Are there any questions before we go? I'm kind of sorry that oh, the block paper isn't on Blackboard. It will be in 10 minutes. Ned Block. Yes, I'll put it up right now. It's an amazing paper by actually a philosopher who gives the best description of heritability explanation that I've read. Although Richard Lewontin also has some good books on the subject. Okay, so I'm going to write that down before I head out. And then if there are any other questions, I'm going to stop recording, but if you have any other questions, I'll hang around for another few minutes.